and the dorsal spinocerebellar fasciculus has entered the inferior peduncle. They form indeterminate fibers, with the exception of a bundle named the fasciculus solitarius, which is made up of descending fibers of the vagus and glossopharyngeal nerves. The transverse fibers of the formatio reticularis are the arcuate fibers already described. The pons. Pons varioli. The pons, or forepart of the hindbrain, is situated in front of the cerebellum. From its superior surface, the cerebral peduncles emerge one on either side of the middle line. Curving around each peduncle, close to the upper surface of the pons, a thin white band, the tinea pontis, is frequently seen. It enters the cerebellum between the middle and superior peduncles. Behind and below, the pons is continuous with the medulla oblongata, but is separated from it in front by a furrow in which the abducent, facial, and acoustic nerves appear. Its ventral or anterior surface, pars basilaris pontis, is very prominent, markedly convex from side to side, less so from above downward. It consists of transverse fibers arched like a bridge across the middle line and gathered on either side into a compact mass which forms the middle peduncle. It rests upon the clivus of the sphenoidal bone and is limited above and below by well-defined borders. In the middle line is the sulcus basilaris for the lodgment of the basilar artery. This sulcus is bounded on either side by an eminence caused by the descent of the cerebrospinal fibers through the substance of the pons. Outside these eminences, near the upper border of the pons, the trigeminal nerves make their exit, each consisting of a smaller mediomotor root and a larger lateral sensory root. Vertical lines drawn immediately beyond the trigeminal nerves may be taken as the boundaries between the ventral surface of the pons and the middle cerebellar peduncle. Its dorsal or posterior surface, pars dorsalis pontis, triangular in shape, is hidden by the cerebellum and is bounded laterally by the superior peduncle. It forms the upper part of the rhomboid fossa, with which it will be described. Structure. Transverse sections of the pons show it to be composed of two parts, which differ in appearance and structure. Thus, the basilar or ventral portion consists for the most part of fibers arranged in transverse and longitudinal bundles, together with a small amount of gray substance, while the dorsal tegmental portion is a continuation of the reticular formation of the medulla oblongata, and most of its constituents are continued into the tegmenta of the cerebral peduncles. The basilar part of the pons consists of A. Superficial and deep transverse fibers, B. Longitudinal fasciculi, and C. Some small nuclei of gray substance, termed the nuclei pontis, which give rise to the transverse fibers. The superficial transverse fibers, fibrae pontis superficialis, constitute a rather thick layer on the ventral surface of the pons and are collected into a large rounded bundle on either side of the middle line. This bundle, with the addition of some transverse fibers from the deeper part of the pons, forms the greater part of the brachium pontis. The deep transverse fibers, fibrae pontis profundae, partly intersect and partly lie in the dorsal aspect of the cerebrospinal fibers. They course to the lateral border of the pons and form part of the middle peduncle. A further connections of this brachium will be discussed with the anatomy of the cerebellum. The longitudinal fasciculi longitudinalis, are derived from the cerebral peduncles and enter the upper surface of the pons. They stream downward on either side of the middle line in larger or smaller bundles separated from each other by the deep transverse fibers. These longitudinal bundles cause a forward projection of the superficial transverse fibers and thus give rise to the eminences on the anterior surface. Some of these fibers end in or give off collateral to the nuclei pontis. An important pathway is thus formed between the cerebral cortex and the cerebellum, the first neuron having its cell body in the cortex and sending its axon through the internal capsule and cerebral peduncle to form synapses either by terminals or collaterals with cell bodies situated in the nuclei pontis. Axons from these cells form the transverse fiber which pass through the middle peduncle into the cerebellum. Others, after decussating, terminate either directly or indirectly in the motor nuclei of the trigeminal, abducent, facial, and hypoglossal nerves, but most of them are carried through the pons and at the lower surface are collected into the pyramids of the medulla. The fibers which end in the motor nuclei of the cranial nerves are derived from the cells of the cerebral cortex and bear the same relation to the motor cells of the cranial nerves that the cerebrospinal fibers bear to the motor cells in the anterior column of the medulla spinalis. Probably none of the collaterals or terminals of the cerebrospinal and cerebrobulbar fibers end directly in the motor nuclei of the spinal and cranial nerves. One or more association neurons are probably interpolated in the pathway. The nuclei pontis are serially continuous with the arcuate nuclei in the medulla and consist of small groups of multipolar nerve cells which are scattered between the bundles of the transverse fibers. The dorsal or tegmental part of the pons is chiefly composed of an upward continuation of the reticular formation and gray substance of the medulla oblongata. It consists of transverse and longitudinal fibers and also contains important gray nuclei and is subdivided by a median raphe which, however, does not extend into the basilar part, being obliterated by the transverse fibers. The transverse fibers in the lower part of the pons are collected into a distinct strand named the trapezoid body. This consists of fibers which arise from cells of the cochlear nucleus and will be referred to in connection with the cochlear division of the acoustic nerve. In the substance of the trapezoid body is a collection of nerve cells which constitutes the trapezoid nucleus. The longitudinal fibers, which are continuous with those of the medulla oblongata, are mostly collected into two fasciculae on each side. One of these lies between the trapezoid body and the reticular formation and forms the upward prolongation of the lemniscus. The second is situated near the floor of the fourth ventricle and is the medial longitudinal fasciculus. Other longitudinal fibers, more diffusely distributed, arise from the cells of the gray substance of the pons. The rest of the dorsal part of the pons is a continuation upward of the formatio reticularis of the medulla oblongata, and like it, presents the appearance of a network, in the meshes of which are numerous nerve cells. Besides these scattered nerve cells, there are some larger masses of gray substance, that is, the superior olivary nucleus and the nuclei of the trigeminal, abducent, facial, and acoustic nerves. 1. The superior olivary nucleus, nucleus olivaris superior, is a small mass of gray substance situated on the dorsal surface of the lateral part of the trapezoid body. Rudimentary in man, but well developed in certain animals, it exhibits the same structure as the inferior olivary nucleus and is situated immediately above it. Some of the fibers of the trapezoid body end by arborizing around the cells of this nucleus, while others arise from these cells. 2. The nuclei of the trigeminal nerve. Nuclei and trigemini. In the pons are two in number, a motor and a sensory. The motor nucleus is situated in the upper part of the pons, close to its posterior surface and along the line of the lateral margin of the fourth ventricle. It is serially homologous with the nucleus ambiguus and the dorsolateral cell group of the anterior column of the spinal cord. The axis cylinder processes of its cells form a portion of the motor root of the trigeminal nerve. The remaining fibers of the motor root of this nerve consist of a fasciculus, which arises from the gray substance of the floor of the cerebral aqueduct, and hence is named the mesencephalic root. It is not altogether clear whether the mesencephalic root is motor or sensory. The sensory nucleus is lateral to the motor one and beneath the superior peduncle. Some of the sensory fibers of the trigeminal nerve end in this nucleus, but the greater number descend under the name of the spinal tract of the trigeminal nerve to end in the substantia gelatinosa of Rolando. The roots, motor and sensory, of the trigeminal nerve pass through the substance of the pons and emerge near the upper margin of its anterior surface. 3. The nucleus of the abducent nerve, nucleus n abducentus, is a circular mass of gray substance situated close to the floor of the fourth ventricle, above the striae medullaris and subjacent to the medial eminence. It lies a little lateral to the ascending part of the facial nerve. The fibers of the abducent nerve pass forward through the entire thickness of the pons on the medial side of the superior olivary nucleus and between the lateral fasciculi of the cerebrospinal fibers, and emerge in the furrow between the lower border of the pons and the pyramid of the medulla oblongata. The nucleus of the facial nerve, nucleus n facialis, is situated deeply in the reticular formation of the pons, on the dorsal aspect of the superior olivary nucleus, and the roots of the nerve derive from it